part of the lab family, and I can't, it's a little bit hard for me to kind of sum up all of the kind of, experiences, but I've just been, the lab has been amazingly fortunate to have you be part of the family, and you'll see in this presentation the hard work she's put in, and some of the things that you probably won't see in this presentation are just the huge amount of mentoring as well that she's done for students in the lab. So we've had a number of undergraduates that have come through, and not only that, but also people that we've been able to help kind of get into this field as well. And so it's been quite a ride. I remember some of the times when we sat in the office and we're doing these thought experiments, kind of thinking about potential outcomes of these experiments, and we had really no idea where it could have gone in terms of the resultant results. But you'll see in this talk kind of the kind of where these experiments went, and they took us in some unexpected directions. And so now, after this, we're going off and interviewing at this top company. So one of the interesting things is we've kind of co-evolved this research with other researchers, Jen Lottwall in Sweden and others, had a chance to go talk there. And so I think what has helped done is actually help really move the field of exosome biology forward. And we've been in these conversations with these leaders in the field, really, and the experiments that Swetha has carried out has actually helped influence them to a certain extent. So anyway, I just thank you so much for all your amazing efforts, and look forward to that.
Thank you, Dr. Weinberg. Good morning, everyone, and thank you for coming out today. I'm going to be talking over the next hour about the crux of my PhD thesis, which is understanding the role of exosomes in innate immunity. Before I delve into it, I want you to imagine that you're in an emergency. What is the very first thing you do? You call 911, and then the first responders arrive on the scene. They assess the damage, limit it, and then they carry you away in these emergency vehicles through the highways. And this is an emergency system that our society has evolved to help react to emergencies. Similarly, our body has an emergency response system as well, and that's our innate immune system. And so similar to the first responders, we have innate immunity, which kicks in right after you face an emergency or an infection. You have these highways in the body, the primary ones of which are blood and lymph. And then you have these vehicles that help carry information from the first responders to these highways. And one of those vehicles is exosomes. And these are the legs on which my thesis stands. And I'm going to talk about each of these in a little bit more detail just now. Let's start with the immune system. So its primary function is to protect us against infection and maintain our health. And it does this using its two arms, innate immunity and adaptive immunity. So imagine you have a viral infection. The yellow curve here shows the increasing virus titer as soon as you've been infected. And then right away, the innate immunity detects it, and then you see the innate immune response peaking. And as a result, you see that the virus titer kind of plateaus out. It's not increasing anymore because the innate immunity has limited its spread. It then informs adaptive immunity of the virus infection, which then starts by producing antigen-specific T cells. And it does this by producing antibodies specific to the virus that's infecting you. And you see that this takes a little bit of time to happen, and that with an increase in antibody, your virus titer goes down and you recover. So these two arms coordinate in a time at different time scales, but the net result is that you recover from these infections. Since it's innate immunity that I'm really interested in, we're going to talk a little bit more about how that kicks into action. So it's initiated when you have an infection. And so imagine you have a skin wound, and then there's a bacteria or a pathogen that enters. This then causes threat detection by specialized immune cells that are present in all tissues. In the case of the skin, it's tissue-resident APCs are a type of cell that will detect this for you. Upon detecting the threat, they will secrete these chemical molecules called cytokines and chemokines, which then results in the initiation of innate immunity. And the key features of innate immunity, as I mentioned before, are they are the very first responders to a pathogen attack, and therefore it's broad and non-specific. And it's also immediate. So if you see the time scale, it's in a matter of hours. So once you have these cytokines and chemokines secreted, they use the highways in our body, which I mentioned briefly before, which is blood and lymph, and they go to areas in the body that are concentrated in immune cells, so spleen and lymph node and other secondary lymphoid organs, where they then inform specialized immune cells, which will then prepare an antigen-specific response, which was corresponding to the virus-specific antibody in the previous slide. And so that's antigen-specific adaptive immunity, but it takes a number of days for that to kick in. So innate immunity contains the first, contains the infection and informs the adaptive immunity, so a specific response can then be, specific action can be then taken. And so I'm only going to focus on innate immunity for the purposes of my thesis. So as you can imagine, the onset of immunity requires a lot of complex coordination between different cell types, and they have to communicate with one another in order to make this happen. And so the classical view has been that the primary messengers of cell-cell communication in the context of immunity are these chemical molecules called cytokines and chemokines. And that was the view for a long time. However, recently, about 10 years ago, it was noticed that all of these immune cells that are involved in an immune response, one, secrete extracellular vesicles, and two, take them up as well. And so the field is now questioning the ability of these extracellular vesicles to be communication molecules or vehicles in innate immunity as well as adaptive immunity. So I want to just talk about the types of vesicles that exist. And so based on their size and their biogenesis pathway, they're classified into a number of types, some of which are shown here. The largest class is microvesicles. They have a size of greater than 1 micrometer, and they're basically just membrane blebs. So the plasma membrane buds off and forms them. The second class is exosome-like vesicles, or as they're more commonly called today, just extracellular vesicles. They have a size of greater than 200 nanometer but less than 1 micrometer. And their biogenesis is unclear, and they don't really have specific markers. The third class is the one that's most studied, and they're called exosomes. And they have a very particular pathway of biogenesis. So they are endosomal in origin. So there are multiple blebs that are found in the endosome called multivesicular endosomes. And these then fuse with the plasma membrane to release these small vesicles contained within, which are then called exosomes. So they are generally less than 200 nanometers in size, and they are very well characterized at a molecular level, and they have a number of tetraspanine surface protein markers to characterize them. And then there is another class of membrane particles that is said to be produced when the cell undergoes apoptosis. For the purposes of my thesis, we only focus on exosomes as the vehicle of choice, as I mentioned before. So, looking at exosomes a little bit more, as I mentioned, their size range is less than 200 nanometers, and this slide shows a more detailed view of their biogenesis. So, you can see these multiple blebs that contain RNA, protein, and microRNA that are found within multivesicular endosomes that fuse and release them extracellularly. Once released, these molecules require some fluid to move to their distal cell where they are taken up. And so they most commonly use all body fluids, and they move distally where they fuse with the target cell and deliver functional cargo. And so they have been pretty well studied, and they have a number of known functions in health and disease. So for example, they play multiple roles in maintaining normal physiology in the brain and the heart. They help in stem cell regeneration, but they're most characterized in the context of cancer biology and infection. And since I'm interested in innate immunity, I'm going to talk about the roles of exosomes that we do know in this context. So exosomes have been known to play roles in antigen presentation. So dendritic cells, which are a class of molecules that present antigens to T cells and initiate specific adaptive immune responses, have been known to secrete exosomes that contain the antigen that can, in lieu of the dendritic cell, present the antigen to T cells, initiating an antibody response, or immature dendritic cells can secrete exosomes with antigens that can then be taken up by mature dendritic cells to present. And there are other roles as well, but the common underlying theme is that exosomes are best studied in the context of adaptive immunity, and their roles in innate immunity are just beginning to be understood. And so this is where I'm focusing my attention. As I mentioned before, exosomes do not act locally where they're produced. They need to travel to distal sites for their action, for them to have some action. And so they do this by using the highways I mentioned before. And so I'm going to talk about blood first. The circulatory system is one of the largest body fluids, and it flows really fast. And when people look for exosomes transport through them, very few people have looked for exosome transport through them, but there was this one study that did some kinetics of transport using the blood. What they did was they produced cell lines with fluorescent exosomes, and then they administered it to a rodent, and then they detected at endpoints the movement of exosomes through the blood. And this is the state of the field that I came into. There were only endpoint studies, and there were very few studies that were actually characterizing exosome transport. The other circulatory system that's really large is the lymph. However, there are no studies that document the transport of exosomes to the lymphatics at this point. And so that is the route of transport that I will be studying in my thesis. So let's look at the lymphatic system in a little bit more in detail. As you can see here, the lymphatic system, much like your circulatory system, is found throughout the body. Its primary function is in maintaining tissue fluid balance, and it also transports lipids. And what's most interesting to us is that it transports immune cells from the periphery to the sites of immune reactions, which are the lymph nodes. Here you can see that lymphatics run parallel to blood vessels. So the one in red is your blood vessel, and the one in green is your lymphatic vessel, and they run parallelly to each other. So if you look at the structure of the lymphatic in a little bit more detail, you see that, like your blood capillaries, you have these initial lymphatics, which are basically penetrating your tissue, and then they converge to form these collecting vessels, which are similar to your artery or vein, and then they flow through lymph nodes in a unidirectional fashion, and they re-enter the circulation of the thoracic duct, maintaining the fluid balance of the body. And so, to come back to my thesis overview, these are the three areas that I am interested in: innate immunity, lymphatics, and exosomes. And the two questions that I'm going to try to answer is, are exosomes trafficked through the lymphatics, and what is the kinetics of this transport, if they are? And two, what are the systemic roles of exosomes in the context of innate immunity? Towards this, I have three specific aims. The first is to establish and characterize exosome transport through the lymphatics in vivo. The second is to show that exosomes play a role in dissemination of innate immune infection using bacterial and viral infection as model systems in vitro. And the third is to tie it all up together and characterize the innate immune response by exosomes in vivo. Let's look at the first aim, which is characterizing and establishing lymphatic transport. And so that will deal with these two arms of my So as I mentioned before, lymphatic transport of exosomes is really uncharacterized. There has been no demonstration or characterization. However, we know that lymph nodes do receive exosomes, because we saw a couple of papers where people inject exosomes and then they see them in the lymph node. And however, the papers did not mention if the exosomes got there through the blood or the lymph, or if there was any contribution of the lymphatic transport. So therefore, my hypothesis was that the exosomes are taken up and transferred by the lymphatics to the lymph node. In order to test this, we need to first establish an in vitro model and verify that it actually gets to the lymphatics. So to come back to the structure of the lymphatics, the initial lymphatics, which are like your capillaries, are formed by the lymphatic endothelial cells, and the cells overlap forming these vessels. And so for exosomes to get transported from the tissue by the lymphatics, they have to enter the lumen of the lymphatics. So we wanted to see if this transport across the cell membrane was occurring. In order to test that, we set up an in vitro system that looks like this. So this is basically a transwell insert upon which we grow cells to separate this compartment into two sides, a basal side and an apical side. And we grow cells on this membrane which make up the vessels of the lymphatics called lymphatic endothelial cells or LECs. And we also want to see if there is a size dependency on the transport. So does this transport only depend on the biophysical characteristics of the particles used? So we used a control of beads, artificial latex beads that were fluorescent, and their size and density matched to that of exosomes. So in order to see if exosomes are transferred from the basal to apical side, we add fluorescent beads and exosomes to the basal side, incubate them with these LECs, and then wait for a predetermined amount of time and check the apical side for fluorescence. When we do this, and so the transport direction is this way. And so when we do this and compare the transport of exosomes and beads, as the size controlled beads that I mentioned before, we see that exosomes are transported 3 times as much as beads in vitro, indicating that it's not only size that is important. We also did another experiment to characterize the role of cells in this transport. So we measured transport in the absence of cells. So basically the membrane was a barrier to diffusion. And then we added fluorescent exosomes and beads and measured transport. And then we added cells and saw how that changed this transport. So here I want you to look at only the dotted lines first. We use 3 molecules. We use dextran in addition to the exosomes and beads I've mentioned. Dextran is a really small molecule, around 2 kilodalton, and should freely diffuse through the membrane and when cells are present between intracellular junctions. So in the absence of cells, the dotted lines indicate the transport and absence of cells. The diffusion of dextran was really high, and here the red line shows the diffusion of beads, and the green is the diffusion of exosomes. So in the absence of cells, the transport is somewhat comparable. But now that we add cells, there is a barrier to diffusion for dextran. So dextran transport comes down, the solid blue line. Bead transport goes almost to 0, the solid red line. But exosome transport goes up, indicating that the cells are playing an active role in pushing the exosomes from one side of the membrane to the other. And so when we take one of these cells on the membrane and look inside it using fluorescent microscopy, so this is the blue here shows the nucleus and the red labels the actin cytoskeleton, so it shows the boundaries of the cell. We see the green fluorescence from the exosomes localized entirely internally. So what this means is that the cells are taking exosomes from the basal side through them and pushing them to the other side, so they're participating in transcytosis. Now that we know that exosomes are being trafficked in vitro, we want to see what happens to them once they get into the lumen of the lymphatics in vivo. In order to do this, I had to first design a labeling system that would allow me to detect these exosomes in real time. So we used a near-infrared dye that is conjugated to the membrane proteins of exosomes, in addition to the green membrane dye that I already added. And this dual labeling system allows us to detect exosomes in animals real time without doing endpoint studies. And additionally, the Dixon lab has a quantitative toolkit to measure the transport through this system. So in an animal model, I would inject these dual labeled exosomes at the tip of the tail, and then we'd focus a near-infrared laser on the base of the tail, roughly 10 centimeters away, and this would be the site of imaging. And then all of these images would be collected by an NIR camera, and then we'd quantify them. And the lymph nodes that drain this are just above the tail, and they're the sacral lymph nodes. And so, if you remember, the lymphatic system is a collection of lymphatic vessels, in the rat tail, which is my model system, you will see two patterns. And so here I inject the labeled exosomes at the very tip, and then I focus the camera here, which is the site of imaging. And so this is a video you're going to see of the mouse dorsally. So the mouse is on the table and you're looking at the base of the tail. So here in this video you're able to see movement of fluorescence across the ends, which indicates that the fluorescent exosomes are moving from the tip of the tail through the lymphatics to the base of the tail. And we see that one vessel has fluorescence before the other, and the vessel in which we see fluorescence first we term the dominant vessel, and the other vessel is termed the non-dominant vessel. As I mentioned before, we have ways to quantify this fluorescence once you detect them. So we chose areas of interest and just measured the change in fluorescence at each of these areas, and we saw that the dominant vessel had consistently higher fluorescence than the non-dominant vessel. And then there was an arrival state where very early the transport was exponential, and then there was a steady state where it had plateaued. So now that we know it's being transported through the lymphatic vessels, the next question was where were they going? So we wanted to look in the draining lymph nodes. As I mentioned before, there are two nodes that drain these two vessels, and they're present in the back of the mouse. And you're again looking at the mouse dorsally. You can start seeing the first node light up soon. And this we term the dominant node, and the node that lights up second is termed the non-dominant node. And then you start seeing the non-dominant node light up as well. And so here are some stills of the same video, just showing that there is a node that lights up first, and that's the dominant node, and here is the steady state of fluorescence. And these lymph nodes are stable retention sites of these exosomes because they are fluorescent even at 2 days post-injection. And this is contrary to the lymphatic vessels, where the transport has cleared out by day 1. So the lymph nodes are retaining it. And the other interesting point is that the fluorescence has not equalized. This asymmetry is maintained even 2 days after injection. Similar to the lymphatic vessels where we quantified the transport through each of them, we also were able to quantify the transport for each lymphatic transport to each node. And here are the transport segments for the dominant and non-dominant nodes. The dominant node always has a higher fluorescence than the non-dominant node in this setup. And we also quantified them at my 2 time points that I used in the study, an early time point of 2 hours and a later time point of 2 days. And this asymmetry was the one I mentioned, it's maintained. So now that we know the lymph nodes retain these exosomes, the next question is which are the cellular subsets that are responsible for this retention? Because the lymph node is a complex mixture of many cell types. So in order to understand this, I took out the lymph nodes, I digested them, and I analyzed the presence of exosomes within various immune subsets within the node. We saw that at 2 hours, primarily macrophages, which is represented by CD11b, which is a macrophage marker, retain exosomes. And at 2 days, macrophages still have a lot of exosomes, but now B cells start showing co-localization, which is represented by CD19b. And we also confirmed this using immunohistochemistry of the whole node sections. So here you see exosome fluorescence within the whole node in green, macrophage fluorescence in red. This is just a merge showing their co-localization. And similarly for B cells, we saw that exosomes were present with them. So to summarize my first aim, this is the first demonstration of lymphatic transport of exosomes, and we showed that the draining lymph nodes stably retain them, and more specifically, macrophages and B cells contribute to a bulk of the exosome capture. This was recently published in scientific reports. The second aim tries to understand the interplay between innate immunity and exosomes, and I use bacterial and viral infection as the models for this study. So coming back to the infection model, as I mentioned, when you have a wound in your skin and you have a pathogen that enters, there are cells that do threat detection and start the innate immune response. They do this using certain receptors on their surface, and these are called pattern recognition receptors. The largest class of pattern recognition receptors are the toll-like receptors, and here I am showing 3 such receptors. TLR3 recognizes poly-IC, which poly-IC is double-stranded RNA that indicates viral infection. LPS is recognized by TLR4, and LPS is a component of gram-negative bacteria, and the last one shown here is PAM, which is a marker of gram-positive bacteria. When each of these ligands binds to the cognate receptor, they set into motion gene regulatory networks that are activated, and a study here shows activation of dendritic cells by each of these agonists, and this is the control, and this is a heat map of the changes of those 3 genes. What you have to notice is that the pattern in the control is very different from the pattern in the LPS or the poly-IC or the PAM. So it's a specific reprogramming and not a general response to infection. So my hypothesis was that the cells that directly are exposed to these infections will then secrete exosomes that can carry the messages of infection distally. In order to investigate this, we designed an experimental setup that looks somewhat like this. So we used control cells, which in my case were K cells, which was an ovarian cancer cell line. And then we had 2 conditions. One was poly-IC, which I mentioned was a marker of viral infection, and one was LPS. We collected exosomes from each of these 3 conditions independently, and then we added them to distal cells, or cells that had never seen any infection themselves, and their only source of information was the exosomes themselves. We then profiled the distal cells using a microarray. We also wanted to compare the response locally, so we took these 3 local cells and then studied them using a microarray as well. So let us first look at what happens in the case of poly-IC. Before I tell you what my data is, I want to explain how poly-IC is detected within cells. So there are 2 modes of detection. One is TLR3, which I have mentioned, and that's endosomally bound. So when poly-IC binds to its TLR3 receptor, it sets into motion a signaling pathway which converges on NF-kappa beta and IRS. It also has a cytoplasmic receptor, and when it binds to its cytoplasmic receptor, another chain of signaling is started, which converges at NF-kappa beta. NF-kappa beta activation results in the secretion of interferon, and this is a hallmark of poly-IC infection. Interferon has its own signaling capabilities as well, and it can act both in an autokine and a parakine fashion. So these are the critical genes that are involved in poly-IC response. And so I looked at both the local cells, which had directly received poly-IC stimulation, as well as distal cells that had only gotten poly-IC, and compared them for the expression of these specific infection response genes. Here you can see a heat map of the gene expression in local and distal cells. The first row is unstimulated cells that were not exposed to anything. The second, column. The second column is cells that were exposed to poly-IC directly. The third is cells that got exosomes from the unstimulated cells, and the last is cells that had gotten exosomes from the poly-IC stimulated cells. As you can see here, both the local and the distal poly-IC are the only groups that respond to this infection, and both of them upregulate the common genes that are responsible for viral response. We also did a pathway enrichment in distal cells. So these are the cells that got either control or poly-IC exosomes. And we looked for which pathways were changing the most after exosome addition. And so we saw that the RIG1-like receptor, which is the cytoplasmic pathway for poly-IC recognition, and the toll-like receptor pathway were both going up. So now that we know that there is a viral response in these distal cells, we wanted to understand the time course of this response. So in order to do this, we compared local cells which were stimulated with poly-IC at these different time points of 0, 2, 6, 12, 24 and 48. And then we compared them with cells that got poly-IC exosomes for similar time points. And then we checked for expression of the genes highlighted in red at each of these time points in the local and distal cells. Here you can see the response of MAS, which is involved in the cytoplasmic recognition, and the solid line indicates the change in gene expression of cells that were stimulated directly or the local cells. And the dotted line shows the distal response or the cells that got only exosomes from these cells. As you can see, both the local and distal cells respond to poly-IC in a similar fashion, but the distal cell response is delayed temporally. So the distal response peaks at about 24 hours. We did this for a number of genes and we saw that the patterns held. Now that we've verified the changes in RNA expression, we want to make sure that this has an effect at the protein level as well. And to do this, we used NF-kappa beta as a model molecule. NF-kappa beta is comprised of two subunits, a P50 and a P65. And when it's active, both of these subunits are in close proximity and they're both in the nucleus, which indicates it's active. And so we use an assay called a proximity ligation assay, which will only show positive signals, in this case red dots, when P50 and P65 are within 10 angstrom of each other. Here we can see the four cell types I mentioned before: local control, which is unstimulated cells; distal cell control, which is cells with control exosomes; local poly-IC cells, direct stimulation; and distal poly-IC cells, which is poly-IC exosomes. The blue here represents the nuclei, and you can see that there is activation of NF-kappa beta in the nucleus, which is represented by red dots, only in the cells stimulated either directly with poly-IC or cells that got poly-IC exosomes. Therefore, we're seeing activation of the poly-IC response at both the RNA and protein levels in distal cells. Similar to poly-IC, we also studied the changes in gene expression distally mediated via LPS exosomes. LPS response, like poly-IC response, starts with LPS binding to TLR4 on the surface this time, and it sets into motion a complex signaling pathway, again converging on NF-kappa beta activation. And it results in TNF secretion. So TNF is the hallmark of LPS activation. And this is the inflammatory phase. LPS is also a highly rapid response and it needs to be shut down because excess inflammation causes tissue damage. And so there's a second response phase to LPS, which is the refractory phase. And this is characterized by shutdown of LPS at two levels. One is TOLIP, which interacts with TRAP and shuts down the response at the very top. And the second is at the NF-kappa beta level, which is mediated by SIRT1. And when SIRT1 activates NF-kappa beta, it can no longer bind and transfer in the nucleus. So similar to poly-IC, we look at the changes in gene expression in local and distal cells of these specific response genes. And here the heat map shows the response genes that I spoke about just now. And similar to poly-IC, we see that the cells that got LPS directly or the cells that got LPS through exosomes are the only ones that are changing their expression for the said genes. Again, I did a pathway enrichment analysis and we saw toll-like receptor signaling go up in distal cells that got LPS exosomes. Similar to poly-IC, I verified the kinetics of exosome-mediated gene expression in distal cells, and we saw again a similar response of local and distal cells to LPS with the accompanying temporal shift in gene expression. We did this for several genes here as well. And we also wanted to verify NF-kappa beta activation, like in poly-IC. So here you can see that when you stimulate with LPS exosomes and you look for NF-kappa beta activation, you see that you can see NF-kappa beta in the nucleus as indicated by the red dots. However, as I mentioned before, when you re-stimulate with LPS, the cells only want to respond once. So re-stimulation with LPS results in them moving to the refractory phase. And as I mentioned, SIRT1 is involved in this silencing at the NF-kappa beta level. And so how it does this is it deacetylates one of the subunits, P65 of NF-kappa beta, and it prevents it from binding to the P50 subunit, and therefore it can no longer be active. So when we re-stimulate cells that get LPS exosomes with LPS, NF-kappa beta is no longer active, and instead you see the P65 subunit now binding with SIRT1, indicating this shift to a refractory phase. So to summarize, it looks like exosomes can transfer the message of infection distally. So poly-IC exosomes induce an antiviral response, LPS exosomes induce an inflammatory response. We also characterize the kinetics of this response, and we show that NF-kappa beta was active in both cases. But now the question is, what about these exosomes is initiating these changes in the distal cells? One of our first hypotheses was that it was because these cells were taking up these exosomes to different levels, or that they had different biophysical or biochemical properties that led to these changes. So we first looked at their overall shape using scanning electron micrographs, and we saw that all of them looked spherical, which is what exosomes are known to be, and they were in the right size ranges. We also looked for these classic tetraspalin markers that define them as exosomes, and two of them are shown here, CD81 and CD63, and they had comparable levels of expression of both these markers. We also looked at the distribution of sizes of these three exosomes, and there wasn't too much of a difference to contribute to this effect that we were seeing. And finally, we looked at cellular uptake, and that seemed pretty consistent across the three cases too. So while we were thinking of what could be causing this, we saw one of these papers where they took a cell line with luciferase, collected exosomes from these called exo-L for the luciferase. They added them to cell lines with no luciferase, and 24 hours later, this cell line was fluorescing. This is a measurement of the change in luciferase activity of this cell line. And they also saw that the transcript for luciferase itself was present in the exosome. And therefore, it seemed like exosomes were doing a good job of transferring RNA from one cell to the other. So our next hypothesis was that the exosomal RNA is what is responsible for the distal effects that we are observing. In order to verify that RNA played a critical role, we used UV radiation that we know causes an elimination of the nucleic acid contents of the exosomes. Here you see three bioanalyzer plots of the size distributions of RNA. The red line shows the native RNA size distribution, and the blue line shows the complete elimination of the RNA within exosomes post-exposure to UV. Now that we have a way to control for RNA content, we then looked at changes in gene expression in a similar way, but by adding exosomes that were exposed or not exposed to UV. In the case of distal exosomes, we saw that almost all gene expression was completely eliminated when we exposed them to UV. We saw a pretty similar effect in the case of LPS exosomes as well, but there were some genes that weren't eliminated, some genes that were still being expressed after UV exposure. We also wanted to verify this at the NF-kappa beta level. And so similar to the previous case where I showed NF-kappa beta activation, in the poly-IC and LPS states, we were no longer able to see NF-kappa beta activation. So therefore, to summarize my findings in this aim, we can say that exosomes can transfer messages of infection distally, and this transfer corresponds to the biological state of the cell of origin. The changes that are induced are RNA dependent, and we can reset these functions that they have upon UV radiation. And we have now established a link between these two circles. This paper is currently submitted. We know that exosomes do use the lymphatics and they are stably retained there. We know that they are able to transfer messages of innate immunity. We want to tie all of them together and see what happens if we're able to initiate an innate immune response using the lymphatics in AIM3. So my third aim is to characterize the changes in immune response at the lymph node that retain these exosomes in vivo. And I did not use both LPS and poly-IC. I only used poly-IC exosomes for this aim. And my hypothesis was that poly-IC exosomes will initiate an innate immune response at the node, but control exosomes will not. To test this, we used the following experimental design. I continued using the mouse tail lymphatic model for injection. So I injected either PBS at the tail tip, or I injected 2 exosomes, so either control or poly-IC exosomes. So there were 3 in total. And then we let the transport take place, and we waited for 48 hours. After 48 hours, we extracted the draining lymph nodes, and then we subject the whole node to an RNA-seq. We then looked for the top chain genes in the nodes themselves. So here you can see a number of genes on the y-axis. This is the absolute gene expression to log 2. The white circles represent PBS or the basal gene expression. The gray is the gene expression induced by control exosomes, and the black is by poly-IC. The first thing to note is that all of these genes are in one of 2 categories. One is their inflammatory genes, and 2 is some of them are involved in neutrophil recruitment. And I will come to what this means in just a minute. Similar to what I've done previously, I also did pathway enrichment, and we saw that all the pathways we saw before, so toll-like signaling, NF-kappa beta pathways, are all activated, indicating that the poly-IC is able to initiate an inflammatory recognition response in the whole node. So as I mentioned, we see neutrophil recruitment and inflammation. What does that mean? So when there is inflammation in the lymph node, the innate immune response kicks in, and it's mediated by 2 primary cell types. One is the macrophages and 2 is neutrophils. And macrophages are present locally within the lymph node, and when they detect pathogens to the lymph, depending on their size, they take them up and they initiate an immune response, which results in neutrophil recruitment. And this indicates that they have recognized there is an infection. And so, since we see that the poly-IC exosomes are initiating both an inflammatory response as well as recruiting neutrophils, we can conclude that they are starting to initiate an immune response against the poly-IC exosomes. We validated some of these genes that I just showed you using qPCR, and as we expected, the control exosomes did not induce a sharp change, whereas poly-IC was able to increase gene expression to a much higher level. We also verified neutrophil recruitment in both control and poly-IC sections, and we used 2 markers, GR1 and Ly6G, both of which are neutrophil markers. And so the first column shows you just exosome expression in the whole node. Here is GR1 expression when the nodes were given control exosomes, and expression when they were given poly-IC exosomes. Similarly for Ly6G. You can see that the neutrophil recruitment markers only go up in the case of poly-IC exosomes. So now that we know the whole node is being reprogrammed, we want to know what happens more specifically. If you remember, I had shown that exosomes are taken up predominantly by macrophages within the lymph node. So we wanted to characterize a more specific response. So we kept the experimental design the same, where we injected either PBS or control or poly-IC exosomes. Waited for 2 days to pull the nodes, but this time we pulled out macrophages from the node specifically, and we analyzed them by RNA-seq. We also did immunohistochemistry of the entire nodes to verify changes in protein expression. And before I show you the results, I want to talk to you about the 2 ways in which macrophages respond to any stimulus. They have two response states called an M1 and an M2. In an M1, when they take up an antigen that is foreign and they need to respond, they go into what is called fight mode, and this is characterized by secretion of multiple inflammatory cytokines. In M2, they have taken up the antigen, but they decided that it's not foreign or it's a self-antigen, and that's a tolerizing response, and it's characterized by anti-inflammatory cytokines. And so, inflammatory gene expression in macrophages has a lot of the same molecules we've seen before, like interferons and the toll-like receptor 3 activation, but it's also characterized by secretion of nitric oxide and IL-1 beta. So when we looked at the pathways that were enriched in the macrophages post poly-IC exosome injection, we saw similar inflammatory pathways go up. So we have NF-kappa beta, chemokine, toll-like receptor pathways, indicating that they were initiating an M1-like response in the macrophages. We also looked for the expression of multiple M1 genes, and similar to the whole node, here you see the baseline gene expression marked by PBS. We see that the control exosomes are pushing them to an M1 state, but they're not doing it fully. It's an intermediate response. But when you give them poly-IC exosomes, this response is completely polarized and it's a stronger response. We also checked for a couple of housekeeping genes as controls and saw that they didn't change in any of the conditions. Similar to whole nodes, we validated the changes in gene expression with control and poly-IC exosomes and saw that poly-IC responded much better than control exosomes for all the M1 genes we checked. And we also checked for NOS2, which is basically nitric oxide and a characteristic M1 marker, and IL-12, which is an inflammatory cytokine in whole node sections. And we saw that both control and poly-IC exosomes here, well, mainly poly-IC exosomes was able to upregulate IL-12 and NOS2, indicating an M1 response initiation in the macrophages that took them up. So, to summarize my findings in AIMS 2 and 3, we know that when cells are exposed to infection, they recognize them using the toll-like receptors on their surface, which they then results in NF-kappa beta migration to the nucleus. They also have the ability to transmit this message through exosomes to distal cells, where they also are characterized by NF-kappa beta in the nucleus, which results in a pro-inflammatory response. When you give these exosomes, and now I mean exosomes from infected cells, to mice in their tails in this lymphatic model, they are transported by the lymphatics to distal lymph nodes. And within the macrophages, they polarize them to an M1 state, which is characterized by NOS2 and IL-12 expression, leading to a pro-inflammatory response. And in the whole node, you see an enrichment of neutrophil recruitment signals, which means neutrophils are coming in, also contributing to this pro-inflammatory response. So, to summarize, we are seeing that one, the whole node is reprogrammed to a more inflammatory state and neutrophils are being recruited. And two, macrophages are being reprogrammed to an M1 state, which tells us that they're getting ready to fight an infection. This is also a part of the same manuscript submitted with AIM 2 to PLOS Biology. And so, my overall summary is that I have established links between the three arms of my thesis. So I've shown that lymphatics transport exosomes. I've shown that they're able to disseminate the message of innate immunity in two model systems. And I've shown a way for the three arms to be linked in the context of in vivo immunity. And so this thesis has resulted in one published paper. We have one paper submitted. Once I leave, there will probably be two more papers, which I might be co-first on. We have a patent application, and I also completed the Tiger program. This wouldn't have been possible without a lot of help from many people, and I want to thank both my labs, the Vanberg lab, which was my home for the past 5 years. I want to specifically thank Sashi and James, who helped me with a lot of the RNA-seq analysis, and the Dixon lab, where I did all my lymphatic work, and a lot of my work in AIM3 as well. I also want to thank my Tiger team, which was such a great part of my experience. And here's my Tiger team, and here's the team that I had for my Copenhagen Business School expedition. My family, who has been my support throughout this journey, and Samit, who has been my family here. My friends who have made this journey so much easier, and my funding sources. Thank you all for your attention, and I'm happy to take any questions.
How does the transport of exosomes relate to the sort of viral replication and the time course of that?
So, exosomes are transported really rapidly. So, as you could see in those videos, you see exosomes in the node like 5 minutes post-injection. So that's really rapid. I think viral replication is also fast, but it's like 20 minutes is the fastest replication that I know of. So what we think is that suppose a cell is infected with a virus that's replicating fast, it's able to send a piece of the virus or some information that a virus is infected through these exosomes. So the medium of rapid dissemination.
And I guess the follow-up question would be the speed of synthesis of exosomes.
So that's a great question. We know that they're constitutively produced, but we don't know the rate, so there's no way to quantify it. I usually collect exosomes after 24 or 48 hours, assuming that's enough time, but given we don't know the rate, we don't know how to quantify that.
This is a naive question, but do you think that where an infection takes place in the body is going to have a meaningful impact on how fast this innate immunity and lymphatic response happens?
So the innate immune system is pretty pervasive,
Right.
And so it's going to kick in no matter where the response is. If it's at a site in the tissue, then the lymphatics will play a critical role in activating the response. But if it's somewhere in the circulation or like in a blood vessel, then it's more likely that the blood plays a bigger role.
I see.
Yeah.
So I really liked your introduction with the first responders and things, but I was walking to work this morning at 7 o'clock and fire engines were going whizzing past with their sirens ablaze. And this happens all the time. They're the first responders. They're massive overcommitment, they're expensive, and they're causing a lot of irritation on the way. Do you have any thoughts about that?
I wish I had a smarter answer to give you than the one going to know. I don't really have something to say about that. But I feel like exosomes are a better emergency response vector because they don't make any noise. So they won't be irritating you.
This is also a very naive question. I'm just curious what you think about it. Like in terms of time course, you're saying the exosomes are getting there much faster than, and in a sense almost prepping some of the B cells long before like a dendritic cell would get there.
Mm-hmm.
How do you think that pre-prepping works? Do you think response would be completely inhibited without that pre-prepping by exosomes? It's not actually activating your T cell.
Yeah, so what we think is that when dendritic cells in the tissue, for example, sense an infection, it's going to take them hours to get out of the tissue into the lymphatics and go to the node and give this information. And they're using exosomes as a proxy for their transport. Instead, they're sending these exosomes to prime the node in a way for their arrival. And it's hard to explain what happens in the context of T cell activation because it takes days. Right? It takes 7 days for you to start seeing antibodies. And we're talking about two entirely different time scales here. This is minutes versus days. So exosomes are characterized to have roles in initiation of adaptive immunity, but it's more in the context of antigen transfer. So dendritic cells can't be everywhere. So they send processed antigen to other dendritic cells using exosomes. Or they want to activate more T cells to produce more antibodies depending on the intensity of infection. So they use exosomes again to send the processed antigen to T cells to activate them. But it's hard to link the smaller time scale to a T cell response, just because they're so different.
I have a question. So you said, so it seems to that you're suggesting that there's some form of active signaling from the dendritic cells to like distal cells, but the alternative interpretation is that they're just secreting exosomes all the time, and it so happens that it reaches the node and they have. So which one of these two models do you think is?
This is a question that's hard to answer because it goes back to the origin of exosomes. Were they a mechanism that was selected because they have these great effects and their ability to move so quickly, or was this a mechanism that existed and was hijacked for information transfer because it was so efficient already? It's hard to give an answer.
So are exosomes better as a prophylactic or as a temporary medication?
I don't think I get your question.
So can the cells remember a particular infection, and can exosomes become immune to that? Like cancer cells can escape the pathway, right? So can exosomes help cancer cells to proliferate?
So I think exosomes are best studied in the context of cancer, and they have documented ways in which cancer cells secrete all of these molecules on exosomes that selectively kill certain immune cell types. For example, natural killer cells are a very strong phagocytic population that goes and attacks tumors in the body. Exosomes have been found that are secreted by tumor cells with a ligand that specifically kills them. And this has been found for several immune subsets. So the tumor does use exosomes to battle and suppress the immune response so it grows better. And it also uses it to seed. So one of the classic papers in the field showed that cancer cells secrete exosomes that go to distal sites and prime them so that when the cancer cell itself arrives, it's able to metastasize better and establish a niche faster. So yeah, in that, it has roles in both.
So can we program them not to do that?
If we could, that would be really great. And that is, I think, one of the areas that many of these companies are trying to enter into as either a cancer diagnostic, that by seeing these particles on exosomes, they're like, yeah, we know there's a cancer that's as yet undetected by other markers, but we can see exosomes with these markers, and therefore it's there, or they want to deliver therapeutics.
Yeah, exosomes also transport prion material. So
Yeah, that's actually very well characterized. It's been shown that, especially in the case of PRP, I'm not very sure what it causes, but I know it's a prion disease.
Right.
They transmit the infective seed form of PRP from cells that have it to cells that don't, and this propagates prions way faster than it would if the cell just had to replicate. And
So we're going to move towards the private phase of it. I just have maybe one public comment, I guess. Some stuff that you didn't show relates to this idea that these exosomes can be loaded and things like that. Do you want to just mention to the overall group about kind of different kinetics that you saw, like telomeres, lymph nodes, the slightly differential kinetics with the poly-IC exosomes?
Yeah, so if you remember, I've characterized exosome transport using the lymphatic system, and I showed that. I'm going to pull up the slide. Give me one moment, please. Have it. So I'm going to just talk about one of these slides for a minute. So if you remember, in the control exosomes, we were able to quantify lymphatic transport at both the vessel and the node level. And when we compare the transport of a control exosome and poly-IC exosomes, you would expect them to be similar. But surprisingly, we saw that control exosomes were transported to a much lower level than poly-IC exosomes in the vessels. And correspondingly, when you looked at the node, poly-IC exosomes were retained much better at the node as well as compared to control exosomes. And when we do a quantification of total lymphatic transport of each of these exosome populations, we see that poly-IC exosomes overall are transported and retained about 3 times as much as control exosomes. And so not only are they causing changes at the node itself, they are modulating the lymphatic transport at a very fast pace to enhance their own uptake and retention as well. And we don't know what exactly causes this, but it's presumably something on the surface. And I think this is an